Have I had COVID-19? Can I get it again? As the first wave of the pandemic slowly recedes, people around the world are waiting to see whether one or more waves will strike. During the corona lockdown, researchers have tried to solve this pandemic puzzle from home. A key safeguard against the second wave is antibody testing. This identifies whether you have had COVID-19, in contrast to virus testing, which only determines whether you have the virus right now. Denmark's largest hospital, Rigshospitalet, University of Copenhagen, and Denmark's largest company, Novo Nordisk AS, have collaborated to develop a unique Danish antibody test.
The overall goal of the project is primarily that we are self-sufficient here in Denmark, that we know every detail about the test, so we are sure that this is high quality, that we can change the test if the virus starts to change, that we can monitor vaccine responses when that becomes available.
We know that when global epidemics, so-called pandemics, hit the world, then there will be limited capacity and there will be a fight for all these commercially available tests. So it's critically important for a nation like Denmark to be able to have unlimited capacity to test its population.
When COVID-19 infects a person's cell, the virus attaches itself to the surface of the cell using a spike protein that is inserted into the cell membrane's ACE2 receptor. Once the virus is attached, the cell absorbs it and is then tricked into mass-producing many more viruses that can invade new cells. The infection has started.
The spike protein is the protein that is crucial for the virus to become infectious. If it was not for the spike protein, the virus will no longer be infectious. So that's why it's very important to concentrate on the spike protein.
The immune system stores information about external threats. For example, the spike protein's unique shape. Then, in a later attack, the body can quickly mobilize its defensive antibodies, which tag virus-infected cells so that they can be destroyed.
You have 7 different coronaviruses. You want to make sure that what you measure is antibodies against this virus, the one called SARS-CoV-2, and not all the others. So you have to have something that is specific.
This specific trait of the virus is called an antigen. The spike protein is a really good candidate.
It does not really mutate. It is critical for the infection of the virus, and it is specific for this particular virus and not found in a similar shape and form among other coronaviruses. So a really good antigen.
But it takes more than a good antigen to make a reliable test. Many existing rapid tests do not appear to be good enough.
They are built up like pregnancy tests, and we have learned now that they are not sensitive enough. They might be used for surveillance of populations to some degree.
Generally, these tests produce too many false negatives. And false positives are even worse, since they incorrectly tell people that they have been infected.
I would not say they're useless, but they are of limited value. They do not always pick up the positive and the negative, so I would shy away from them.
The new test uses a different strategy.
This so-called sandwich ELISA is built on that we have the viral antigen coated in a plastic well, and very, very shortly we add serum, and if this serum contains antibody, it will bind.
Only antibodies developed specifically against the spike protein will bind.
The plates are then washed, and we add in the same antigen, but then this antigen is coupled to an enzyme that can bind to the other arm of the antibody, and this can be developed and makes a color, and then we can see whether there is an antibody reaction against the virus.
We've been able to test out all kinds of sera from minimally to maximally infected patients and identify that it picks up with sensitivity, the fact that you actually are positive, and with specificity, meaning the risk of actually having a false positive.
The test is a world-class test. It seems that it's almost 100% specific.
Due to the severity of the COVID-19 pandemic, having definite knowledge about the antigens that the tests use and about the quality of the tests is crucial. This is why the new tests are built from scratch and based on high-quality viral protein.
If the virus really changed, we could also very easily change the antibody test by following how the virus develops.
The Danish researchers have also decided to develop a new quantitative antibody test.
We can also measure the amount, how it will change over time. And all these things together will give an impression how immune the particular individual is against the virus.
The collaboration between Rigshospitalet, University of Copenhagen and Novo Nordisk AS has been a perfect match.
Public-private partnerships are critical. The public cannot produce a cure. The public cannot produce a vaccine. The public cannot produce this high-throughput testing that Novo Nordisk has done. But we as companies, we cannot take care of patients. We do not have the hospitals and the patient insights that they do. So there's been a high amount of mutual trust. It's been a seamless collaboration, and this has been done in a matter of 2 months. We just want to help for the sake of human health. We want to help the public get over this crisis.
The final trials of the test have just ended, and the results are so convincing that it's possible to start testing up to 5,000 people a day. However, a decision on how the new test will be utilized has yet to be taken.
Then it will be critically important not only that we can test the Danish population, but that we can have done it over and over again. You want to follow the development of the epidemic, both at the population level, but you also want at the individual level. You would like to know, have you had the infection? So can you go and visit your old grandmother because you've had it and you're immune and you will not infect her? If I do not have antibodies and I'm at risk because I have diabetes, I have hypertension, I have had lung disease, then it's important to be cautious. We can ensure a safer reopening of the society and monitor what's going on and try to avoid the hardest impact of a potential second wave.